Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker Anthony, scientific editor of the Molecular Therapy family of journals. This episode features a conversation between the editor-in-chief of Molecular Therapy, Dr. Joseph Glorioso, and Dr. Kyriacos Mitro from Oxford Biomedica and Rachael Nimmo, formerly from Oxford Biomedica. They will discuss an article recently published in Molecular Therapy by Nimmo and colleagues titled Efficient in Vivo Generation of CAR T Cells Using a Retargeted Fourth-Generation Lentiviral Vector. If you're as fascinated by gene editing as we are, you won't want to miss our upcoming event, Breakthroughs in Targeted In Vivo Gene Editing. This is a chance to engage with the latest breakthroughs and take practical knowledge back to your own work. Join us in San Diego or virtually, November 20th to 21st. Check out the speaker lineup and register at asgct.org/breakthroughs. And now, Drs. Glorioso, Mitrofanous, and Nimmo.
Hi, Kiara and Rachel. I'm really happy that you're here today and to talk about your exciting work and how to transduce and make CAR T cells in vivo, which is a very exciting and important area of research. Along the way, we'll just ask you a few questions if that's okay. And one of the things that I was thinking about, just to introduce our audience, for some folks who may not be that familiar with this, that CAR T development began, I think it was the first approval was 2017 by the FDA. And since then, a tremendous amount of progress has been made. So maybe you guys can, relate a little bit about the history of CAR T cells and what do you think about the future?
Great. Thanks for inviting us onto the podcast, Joe. And we're looking forward to the discussion of this exciting topic. So let's give a little background. So my name is Kyriacos. I'm the Chief Innovation Officer at OXB, and I've been working on lentiviral vectors for over 25 years. So I joined the Kingsman Group in Oxford University in 96 to develop lentiviral-based vectors. And from that group, OXB was spun out, and I joined the company in 97. And we were the first group to develop lentiviral vectors that were clinically relevant, so minimal genes incorporated from the parental viral system. And we were able to develop a manufacturing process, obtain safety and efficacy data that allowed us to initiate the first clinical trial of a directly administered lentiviral vector in 2008 for Parkinson's disease. Now, the attraction of lentiviral vectors is based on their ability to deliver genes to both dividing and non-dividing cells. And because they integrate into the target cell chromosomes, the modification is permanent and leads to stable long-term expression for many years. For in vivo use, we had to develop highly concentrated vector with the appropriate purity. And this meant a lot of effort over the years, improving vector design, developing manufacturing processes, and purification to give appropriate titer and purity. In 2014, we partnered with Novartis to supply them with lentiviral vector for their autologous CAR T therapy that they had licensed from UPenn, which became the world's first commercially approved, as you mentioned, in 2017, first commercially approved lentiviral vector containing therapy Kymriah. Now, this started the journey of the company to becoming a pure play CDMO in viral vector production. And we now have facilities in the US, Europe, and the UK, and specialize in viral vector production, especially for AAV and Lenti. Because of our deep understanding, the considerable work on lentiviral vectors, we feel that we are uniquely placed to develop these vectors for in vivo use. to realize the full potential of CAR T technology. I'll hand over to Rachael to introduce herself and maybe further describe the work we have done.
Thanks, Kiri. And yeah,
Thank
thank
you.
you, Joe, for the invitation to speak today. So my name's Rachael Nimmo. I was the director for the cell technology group at OXB, where I was leading the work that was being done on developing technologies aimed at enabling the next generation of cell and gene therapies. And we were particularly focused around the development of an in vivo CAR T cell therapy. So my team performed the work that we describe in the paper on the development of an efficient, retargeted fourth generation lentiviral vector that was recently published in Molecular Therapy, and I'm a co-inventor on the patent that resulted from that work. So in that paper, I just thought I'd give you a quick summary of what we showed, and that was that we described an improved, retargeted NIPA envelope system that overcame the hurdles of some of the previous vectors by improving potency, and combine this with a fourth generation lentiviral vector platform called the tetravector system, which has enhanced quality and safety. And we've shown that these vectors generated with the system were capable of highly efficient and specific T-cell transduction in vivo. And this led to the generation and expansion of large numbers of CAR T-cells, which then went on to induce a rapid B-cell aplasia. So we were really building upon previous work that had been done by Christian Buchholz's team, who had developed this NIPA retargeting system, but we made some significant improvements to that efficiency by altering the composition of the envelope, so that we incorporated both the retargeted and a non-targeted NIPA attachment protein. So in addition to this, as I mentioned, we utilized a novel fourth generation vector system, and this is anticipated to have an improved safety profile. This is because it has modifications to the viral genome that eliminate the apparent splicing that can happen with these vectors, improve the transgene transcriptional insulation, and substantially reduce expression of the CAR during vector production, which is really important to stop that CAR from being present on the vector surface. So together, I think that these improvements are really going to be important to be able to enable the implementation and feasibility of an in vivo CAR T-cell therapy approach, which we hope will then improve access to these types of curative therapies for patients.
Yes, so I have a couple of questions. One is that you've tackled one of the most important issues, which is the cost of ex vivo development of CAR T cells and having to extract T cells from the patient and transduce them in vitro. Obviously, transduce them in vivo is a huge important step forward. And I particularly am excited about the Nipah virus approach because you can target very specifically to CD3 and CD8 as you show in your paper. And that's because there's a two-step virus entry process where it has to bind to a target that then the G protein, which is I think has been modified with a DARPin or just edited so that it recognizes CD3. And then it activates the F protein, which is true in a lot of different viruses, which then cause fusion. One of the interesting questions here is, and I think you addressed this in your paper, is how do you avoid off-target transduction, like in the liver, for example? And I think you nicely showed that you can avoid that by having this kind of targeting. One other issue that I was interested in was that since you're targeting T cells in general, there's been a lot of interest in dual targeting with the TCR in order to, if one antigen disappears, maybe you can hit a second one. Have you considered doing that in your work, or how you address that problem?
Maybe I could take the first question around the targeting, and then Kiri can maybe address the second one. As you mentioned, it is really important to be able to efficiently target the vector particle to the cell of interest. So in this case, the T cell, both for safety and efficacy reasons. So there's two main ways that people have been doing this. One is using an envelope system from paramyxoviruses like measles or Nipah, or from the VSV virus, or from the related Coccal virus. So we didn't know which one of these systems would be the best one to use. So we initially started to develop targeted vectors using both systems. But there are some key differences between the two that's worth highlighting. The nipper envelope system relies on two proteins, which is a G protein and an F protein, as you mentioned. So the G protein mediates the attachment and the F protein mediates the fusion. And whereas for VSBG, both the binding and the fusion are mediated by a single polypeptide chain. So the first thing that we need to do was to prevent the envelope protein from actually binding to its cognate receptor. So that can be challenging to do with VSBG because there's this trade-off between achieving the full detargeting that you need to achieve and the efficiency of the fusion being maintained. So we reasoned that this was actually going to be easier to do and more effective with the nipper system because of the fact that the binding and the fusion are on separate proteins. We were sort of, as I said, building on work from Christian Buchholz and others who'd first identified the key residues that were required for binding to the cognate receptor, which is the ephrin B2 and B3 receptors. They'd already showed that mutating 4 of these residues together could ablate the binding to the natural receptor, and that truncating the protein enhanced its ability to pseudotype the vectors. So then to retarget those vectors actually to T cells, what we've done is then to fuse a targeting moiety such as a single chain antibody or a DARPin that has specificity to T cell antigens such as CD3 or CD8 on the surface. So the big issue that we had initially was that the functional titers of these vectors was very low, despite getting very high numbers of particles being produced. So we sort of looked to the previous structural and mechanistic studies that had been done on nipper viral envelope fusion, and this led us to an idea for how we might be able to optimize the efficiency of the vectors. So the G protein is actually made up of a tetramer. So we hypothesized that only one of those subunits within that tetramer would actually need to display the binder in order to mediate binding to the target cell. And that by having a mixture of retargeted and non-targeted blinded G protein within that tetramer, that we could actually reduce the steric hindrance that might be happening due to the presence of the binder on the G protein, while retaining the ability of that protein complex to bind to the target receptor on the T cells.
That's great. I love that.
Yeah, it was great. And it actually worked. I mean, that was the most amazing thing. Normally you have a hypothesis, it doesn't work. But yeah, in this case, it was very successful. And across a range of different ratios of the retargeted and non-targeted G protein, we saw a significant improvement in transduction efficiency. And we called this a mixed envelope just for the ease of use in terms of terminology. And we saw a several-fold higher titer compared to vectors that were pseudotyped with an envelope that had only the retargeted G protein. We also found that the use of the DARPin with the mixed envelope resulted in around a 50-fold increase compared to the original vector that we'd been using with the SCFV binder. And these were able to specifically and efficiently generate very high numbers of CAR T cells in vivo in a humanized mouse model and to successfully ablate target B cells. And there was very minimal off-target transduction observed. This was unlike for the wild-type VSBG, which we used as a control. This, where we saw fairly high levels of vector actually being detected in the liver, we saw very little with the nipper system. So we think that improvement in transduction efficiency combined with the specificity is really important, as it means that we can increase the number of doses that we might be able to generate from a single manufacturing batch. That will be obviously critical for then the cost of goods for these vectors going forward.
Fantastic. So I guess you also said that it was a TCR activation independent. I get you need that for ex vivo CAR T production, right? And so, but in vivo you didn't need that. So that's a really another big step forward, I think.
Yeah, that was quite surprising because we know that T cells do have restriction factors that limit their transduction when they're quiescent or non-activated. But actually, we found that using a CD8 targeted lentiviral vector, where we weren't actually triggering the TCR, that we still saw very high levels of transduction in vivo and in vitro. So, it doesn't seem to be necessary for that to occur.
Kyria, how do you, what's the best way to deliver an in vivo targeting vector like that? Is it in the tumor or in the IV in the blood? How would you see that?
Yeah, so the way we're looking at this would be that this would be an IV administration. The vector, as Rachael described, is specifically targeted to T cells. So we would envisage administering into the bloodstream. There are T cells there. Because we are using the Nipah system targeting CD3, the cells do not have to be activated, but anyway, we think around 10% of the cells may be in an activated state in the bloodstream based on various publications. And then we would envisage that the vector would bind through the modified Nipah system and allow transduction of those cells. One of the key technologies that we have employed here is to use something called the TRIP system. So there's this aspect of directing the vector particles to the T cells, but we know that during vector production, you can get large amounts of the CAR protein generated, and that CAR protein can end up on the vector particles. And this may allow, in rarer cases, but it's an issue when you're thinking about treating many patients, allow binding to, let's say, CD19 on cancer cells or autoreactive B cells. So one of the technologies we developed is the TRIP system. This is a way of preventing translation of the CAR protein, so no CAR protein during production, so that the vector particles, when administered into the body, they don't have CAR on the surface. So the only binder is that to your T cells, and this we think it will be important for ensuring that we're targeting the right population in the body.
Fantastic. So one of the things that always comes up in these kind of discussions with CAR T cells is, will this approach be useful for treating solid tumors? And what do you think about the entry of, say, for example, your engineered cells in vivo? Will they be able to enter into, say, melanoma cells or breast cancer cells?
Yeah, maybe I can start and then Rachel can jump in. So the way we envisage this, there are a broad number of advantages to in vivo CAR T. It's going to be a single off-the-shelf treatment. This means that patients can be treated soon after diagnosis. You won't have to take the cells out, you won't have to process them, and there is limited capacity anyway in terms of GMP cell manipulation. We think about 100,000 patients have been treated so far with CAR T therapy, even though many more patients would have benefited, but because of the limited facilities and having to treat patients sometime after diagnosis, it limits the possibility of using this technology. If we move to in vivo CAR T, we can treat patients sooner. There will be no need for lymphodepletion. The patients are likely, a broader patient population would be eligible for treatment because you don't have to have lymphodepletion. And because CAR T cells are generated in the body and not ex vivo manipulated, they're likely to be more efficacious. We'll have to verify that in clinical trials, but that's one of the things that would be interesting to see. Now, this technology that we describe in this publication is a way of delivering genes. The vector system that we've developed, as Rachel mentioned, is this fourth generation vector system where we've removed unnecessary sequences from the vector. We've removed splice acceptors, splice donors. We've added in additional safety sequences within the LTRs to insulate the vector from the surrounding DNA and also insulate the surrounding DNA from the vector. And this gives us the capacity to deliver multiple genes. So when we think about solid cancer, we may need to deliver 1, 2, or 3 different genes. And with these vectors, we can do that. We can also think perhaps of screening a cancer for which antigens are present. And maybe we can administer a cocktail of targeted vectors that would generate the right population of CAR T cells in the body. So instead of having to do CD19 and then autologous CD19 targeted CAR T, and if that doesn't work, you then switch to BCMA, for example, for lymphoma. Maybe you can do that at the same time, and maybe we can apply that in solid cancer. Maybe we can engineer the CAR T cells to express various cytokines that would allow increase the chance of efficacy. So we're essentially relying on the the research that's going out there in solid cancer, and we are providing the means of delivering the successful set of genes that will have been identified by others, as well as work that we've done into how to achieve successful solid cancer therapy. But I believe that won't be far off now. But Rachael, anything else to add to that?
Yeah, I suppose actually also the fact that we don't have to do lymphodepletion is going to be quite a big advantage for solid tumors as well, because it means that the bystander immune cells can actually get more involved in that tumor targeting, and so actually may be more efficacious. But that's obviously speculation until we actually see the data coming out from the trials.
This technology provides the opportunity to treat first rather than last, right?
Yeah.
And that's a very important step because, after you've treated, say, leukemia for a while, you lose a lot of targets and so on. And so this may avoid those kinds of problems. Do you know how long these cells endure after they've been transduced, or is that something you've been looking at?
We don't have data from in vivo CAR T studies, but we have data from other in vivo clinical trials that we have done for the eye and for the brain. And we know that we can, once the cell has been genetically modified, the expression, depending on the promoter you use, can last for many years. From autologous CAR T therapy, we know that CAR T cells are present for many years, so over 10 years, I think. And they're in the background. If B cells reappear, they seem to be killed off again. So the CAR Ts are present, and there is that stimulation. Our expectation is that with lentiviral vectors, the advantage is that they integrate, the cells divided, you keep that modification. We would expect that they would last for the lifetime of the patient. But yeah, we'll have to verify that in clinical trials, but there's no reason to expect that they would have a different lifespan versus autologous CAR T. Maybe it'll be even better given you haven't had to do the ex vivo manipulation, but we'll have to see.
I see. So this is something I was wondering about is, do you think that this strategy will be useful for treating autoimmune diseases, which there's a lot of interest in that?
Yes, so there are very exciting data coming out from various clinical trials using autologous CAR T. We believe that the in vivo CAR T with lentis will be able to make a big difference there. Rachael, do you want to talk a little bit about that in terms of the
Yeah, I mean,
history?
I think there's been some really exciting work done recently, hasn't there, with by especially George Shett and others, who've shown that the CD19 targeted CAR T cells can also be used to treat a variety of autoimmune diseases by eradicating those autoreactive B cells. And I think the results from those initial trials have proved really very compelling and revealed a very profound reset of the immune system in many of those patients. And they appear to be achieving complete remission. But obviously, this will need longer term follow up to really understand how durable those responses are. But I think obviously that means that you're increasing the number of patients that could be potentially treated with CAR T cells. And this is where there's a critical problem, right? Despite the transformative potential of CAR T cells, many eligible patients, I think it's been estimated up to 80% are not able to actually receive commercial CAR T cell treatment due to, variety of issues associated with cost and logistical complexity of making these bespoke therapies. So I think these issues will be even more problematic potentially as CAR T therapy looks to move into earlier lines of therapy and to larger indications such as autoimmune disease. So this is where I think, an alternative method for generating the CAR T cells, as we're proposing as an in vivo approach, would really, open up accessibility to these therapies for many more patients and really help to realize the potential of this type of therapy.
Well, the beauty here is that you can really democratize this by providing the vector, worldwide or something, so that once laboratories or physicians are trained to use it, which is not very complicated here because it's delivery through the venous system. It just opens up the door for amazing changes in the application of this kind of a treatment. The other thing I was wondering about is that do you run into issues with exhaustion, T cell exhaustion? Do you have to arm these CAR Ts, say with IL-18, or what are your thoughts about that?
Rachel, I think that's one for you.
I mean, I guess there's not been enough clinical trial data amassed yet. I mean, it's only recently that we've seen data coming out from the trial that ESA Biotech have been doing in myeloma patients, and they've found, that they've got very good responses in those patients, including 2 complete responses with a relatively low dose of vector. Whether or not those responses will be maintained over time, and what the durability of those responses is will be something that would need to be determined. And then I think that's at that point we could, look at whether or not any additional armoring or kind of expansion switches or other types of methods for preventing exhaustion are actually needed. But it may be that you don't want the CAR T cells to be kind of too active for too long. So it may not be something that's desirable from that perspective.
Do you think you can repeat dose the vector in patients, or maybe that won't even be required? I don't know. Do you get immune response to the virus is what I mean.
Yeah, so our expectation is that what we're aiming for, given the way we've designed the purity, the quality, and so on, is that we will not need to redose, but there is the formal possibility of redosing. You will get antibody responses, I would envisage, but there are ways to dampen those down if needed for a second administration. But I think we will not need to redose given the degree of efficacy we've seen. And if we do, it may be some time after the first administration. So, yeah, we will have to see. I guess the right answer to your question is we don't know. But we'll have to see if that's necessary in the clinic. There is the possibility of using a slightly different envelope in due course if there is an issue. But because you're putting in a bolus of vector, if there are any antibodies or in the body from the first administration, you probably would squelch those out and end up with generating the CAR Ts anyway a second time.
Right.
I think the better approach is that of looking at the cancer cells, understanding what the antigens are present on those cells, and in that first administration, giving the right set of CAR T generating vectors that will prevent the need for a further treatment. But we'll have to see if we can redose.
That's very exciting. Wow. I guess I've run out of questions. I just want to congratulate you on such amazing work, and it really represents the future. It's very, very exciting. And I guess this is going to be a real boon to immunotherapy. And I think as time goes on, maybe we'll have CAR Ts that'll open up the tumor and then allow other CAR T cells to enter the tumor, like in the case of solid tumors. So, or using oncolytic viruses or something. But I think that some tumors are very, very difficult because they're so hard, like pancreatic cancer and so on.
Yeah.
But I think that we can't at the moment we can't envision curing everything. But I think the really common cancers, including skin cancers, even brain tumors, perhaps a biggie, of course, is breast cancer. And so I think this is really an exciting development, and I wish you all the best, and hopefully it continues to work.
Thank you very much. Yes, we share that. We share that sentiment. CAR T has shown amazing efficacy over the last a few years, and it would be good to see it being available to more patients. It would be good to see that the cell populations that can be targeted can be expanded. And by providing a means of delivering those genes more affordably to more patients sooner in the patient's cancer, then the more likely we'll have efficacy, the more likely we can treat more conditions. Getting investment into this kind of research is a challenge, but if you can get rid of that issue of having to manipulate the cells ex vivo, that costs a lot and is limited in its capacity, is likely to broaden the amount of research that happens, because you can see an application for it in the future to treat more patients. And from my understanding, the number of autoimmune patients versus cancer patients, it's 10 times higher. So we're very excited about the prospect of this being used for rheumatology and neurology, as well as solid cancer in the future, as you mentioned. So an exciting time for the industry, I think.
So just one last issue that I was wondering about is that the transduction into CAR cells event, that's TCR agnostic. Is that how that works? Or?
Yes, it'll transduce. The vector will go through any, for example, CD3 expressing T cell, and express the CAR that we've engineered in. Maybe, as you mentioned, multiple CARs if necessary, or other capabilities can be added in that can increase the chance of efficacy. There's many trials out there showing that additional cytokines and or dual CARs are beneficial. So this technology allows you to take advantage of that to treat more patients and maybe earlier in disease.
Right. Okay. Fantastic.